So, good morning, students. Today's topic for our lecture is metabolic derangements in diabetes mellitus. Myself, Dr. Hasit Lad. Like you all know, before starting any lecture, we should be aware what we are going to learn. So, what are the objective of today's topic that we should know? At the end of the lecture, student must be able to define what is diabetes mellitus, should be able to compare and contrast the effect of insulin and anti-insulin hormone on metabolism of various macromolecules like carbohydrate, lipid and protein, should be able to describe the metabolic derangements in these macromolecules. So these are the objectives under which we will learn today's topic. So by now you are all aware that what is diabetes mellitus. What is diabetes mellitus? It is a metabolic disease caused by absolute or relative deficiency of insulin leading to hyperglycemia. Diabetes having two types, type 1 and type 2. Type 1 is due to absolute deficiency of insulin, whereas type 2 insulin is there but it is not able to act. So that is due to insulin resistance. In the previous lecture of regulation of blood glucose levels, we had insight into the effect of various factors which maintain the blood sugar levels or blood glucose levels. In that we have effect of insulin on maintenance of blood glucose and effect of anti-insulin hormones on maintenance of blood glucose levels. Both of these opposing hormones tend to maintain the blood glucose in a narrow range very efficiently. So to understand the metabolic derangement, we are just brushing up through the effect of insulin and anti-insulin hormones on blood glucose levels so we can understand in a better manner, better way. So what does insulin do on metabolism of various macromolecules, especially glucose? Insulin promotes glycolysis, glucose utilization via glycolysis and formation of or synthesis of glycogen from glucose, that is glycogenesis. So it reduces blood glucose levels. At the same time, it also inhibits the factor which increases the blood glucose, that are it inhibits gluconeogenesis and glycogenolysis or glycogen breakdown. So net effect is reduction in blood glucose level. If you see the effects of insulin on lipid metabolism, it favors lipogenesis, it's tend to synthesize lipids. It is anti-ketogenic in nature and it inhibits lipolysis because glucose is already there for generation of energy. So there is no need of lipid to undergo breakdown and generate energy. So it inhibits lipolysis. It also inhibits the catabolism of proteins, amino acids, especially we all know that glucogenic amino acid tend to generate glucose via gluconeogenesis. Again, the same concept, already glucose is available, we don't need other sources of glucose for utilization. In contrast to the effect of insulin, what does anti-insulin hormone do? They tend to increase blood glucose level. How do they increase blood glucose level? First and foremost, they have opposing action of insulin. So they tend to inhibit glycolysis and glycogen synthesis. They also directly increase blood glucose by promoting synthesis of glucose, gluconeogenesis, and also promoting breakdown of glycogen stores in liver. This set of hormone has opposing effect also on lipid metabolism, where it inhibits lipogenesis, at the same time favoring lipid breakdown, that is lipolysis, and also they are ketogenic in nature. They tend to favor the synthesis of ketone bodies. For amino acid, they stimulate the catabolism, protein and amino acid, and tend to thereby it generates glucogenic amino acid, which contributes to maintain blood glucose level. So these are the effects of insulin and anti-insulin hormone on metabolism of various macromolecules. Normally, what do they do? That we are aware now. So, after studying these contrasting effects, now we will be able to understand the metabolic derangement due to defect in insulin secretion or defect in insulin action, that is due to diabetes. So, what contributes to the metabolic derangement in diabetes? It is the high glucagon to insulin ratio, which is the culprit for all the derangements observed in diabetes mellitus. So first we will have insight into the derangements in carbohydrate metabolism. Since diabetes is an insulin deficient state or insulin resistant state, the effect of insulin won't be observed. So there is a decreased uptake of glucose into the tissues. So there is more glucose in blood. There is also inhibition of utilization of glucose via glycolysis. So there is defective uptake and inhibition of utilization. At the same time, there is also stimulation of glucose synthesis. So all these factors leads to net effect of hyperglycemia. So there is defective or decreased uptake of glucose in the tissues and utilization of glucose in the tissues. So the glucose remains in blood, and also there is stimulation of glucose synthesis in liver. So it also contributes to blood glucose. So there is a net effect is hyperglycemia. Now these metabolic derangements are responsible for various cardinal symptoms that are observed in diabetic patients. Which are those symptoms? So that are polyuria. Now glucose in diabetes mellitus due to defective insulin action or deficiency of insulin, there is a rise in blood glucose level gradually, and we all know once the glucose passes through the renal threshold level, the glucose rises more than 180 mg per deciliter, it begins to appear in the urine. Now here glucose being an osmotically active molecule, it also draws water along with it. So there is a excess loss of water in urine, so that leads to polyuria. Now since there is an excess loss of water from the body, what happens is there is activation of thirst center present in the brain, precisely in the hypothalamus, that gets activated and it compels the individual to have intake of more water. So there is increased intake of water, that is known as polydipsia. And if you observe the diabetic patients, especially type 1 diabetic patients, The body, due to defective glucose utilization, there is a breakdown of lipid stores in the body, especially the adipose tissues, and also after that there is a breakdown of proteins, especially from the muscle proteins. We get glucogenic amino acid to get the energy. So there is a catabolic state, there is a breakdown of fat storage, protein storage. So that leads to weight loss and muscle wasting. Due to this, patient perceives that he or she is losing weight and they tend to eat more. So that leads to polyphagia. So these are the three cardinal symptoms that are observed in diabetes mellitus: polyuria, polydipsia and polyphagia, which are linked to the derangement in the metabolism of macromolecules. So after studying derangement in carbohydrate metabolism, we move on to derangements observed in lipid metabolism. Now, since the body and the tissues are not able to utilize glucose, they will need energy, and in these conditions, the fat or the triglyceride act as alternative source of energy. So what happens under the hormonal effect? Anti-insulin hormones, they activate lipolysis, and it leads to breakdown of fat stores and generate free fatty acid, which are delivered to the liver. So ultimately there is high free fatty acid in the blood that are observed. Also more amount of fatty acid that are deposited in the liver, which leads to fatty liver, and few of fatty acid molecules from the liver is transported as VLDL micromolecule, VLDL lipoprotein. Ultimately it goes to the tissue and provides fatty acid, forms IDL, LDL. So it all these things leads to hyperlipidemia also. So this lipidemia is observed in diabetic patients due to fat mobilization, and ultimately from the fatty acid, the 2-carbon compound that is ultimately produced, that is acetyl CoA, which undergoes TCA cycle for energy purpose. So increased acetyl CoA, which is meant to generate energy via TCA cycle, but it is diverted to ketone body synthesis, which acts as an alternate fuel. So why acetyl CoA here is getting diverted for ketogenesis, that we will understand. So lipolysis produces excess of acetyl CoA, and this acetyl CoA undergoes TCA cycle combustion and produces energy or ATP with the help of oxaloacetate. Now there is a problem in diabetes mellitus that concentration of oxaloacetate is limited, because oxaloacetate in diabetes will be diverted for synthesis of glucose, that is gluconeogenesis. So oxaloacetate via the 4 irreversible reactions we know, the malate dehydrogenase reaction and phosphoenol pyruvate carboxykinase reaction ultimately generates pyruvate, and ultimately pyruvate leads to synthesis of glucose, which provides glucose to the tissues. So there is less concentration of oxaloacetate and high concentration of acetyl CoA. So there is impairment of acetyl-CoA utilization via TCSI. So where does this excess acetyl-CoA gets diverted to? It gets diverted to since it is a starting material for ketone body synthesis. It goes for synthesis of ketone bodies, which are also an alternative fuel to glucose to certain tissues. So these are the biochemical basis which leads to ketogenesis in diabetes mellitus, especially type 1 diabetic patients. After having insight into the derangement into the lipid metabolism, we'll see which are the metabolic derangement observed in protein metabolism. In diabetes, again the glucose is not available for utilization, so body protein stores undergoes breakdown because it yields glucogenic amino acid, and from glucogenic amino acid we can synthesize glucose in the liver, and this could be delivered to the various tissues. So there is a protein breakdown for gluconeogenesis, which ultimately leads to muscle wasting. So these are the various effects that are observed metabolically in a patient of diabetes mellitus. So if you summarize the metabolic derangements that are observed in diabetes mellitus, so diabetes due to deficiency of insulin, or it is due to insulin resistance. There is deranged carbohydrate metabolism in diabetes resulting in hyperglycemia. There is also derangement in fat metabolism that results in increase lipid levels in blood or dyslipidemia, and also derangement in amino acid metabolism ultimately leads to depletion of protein store and muscle wasting. I hope this video or voice over PPT is informative to you all, and also I hope the previous lecture helped you in learning about regulation of blood glucose. Kindly give your suggestion, kindly go through these lectures, and if you have any query, kindly put it across YouTube so I can solve those queries for you, and do share your feedbacks also. Thank you.